Hello. Ehm, så när vi gjorde labdemonstration på söndag på kursträffen i digitala kursstart. Jag gjorde ett misstag. Jag glömt att spela in demonstrationen. Och jag ber din ursäkt om det. Men jag tänkte jag kan spela in igen nu och göra om demonstrationen och den viktigaste delar. Och så att jag är konsistent. Jag måste säga, jag bytte det över till engelska på söndag afton. Så jag tänkte jag kan göra det nu. Så jag är säker jag säger samma sak. Och precis när jag är klart jag kommer att ladda upp på Canvas på nätet så ni kan gå igenom filmen igen. So byter vi över. Thank you. And the point of this demonstration today, or on Sunday, was to basically have an informal set of demonstrations mainly associated with microbiological methods around culturing and microscopy. We went over very little about molecular techniques because they're not really very demonstration worthy. They're a little bit on the boring side because it's just a lot of pipetting. So it's kind of hard to demonstrate that in a really pedagogic way. But we can go through certain things about culturing, enrichment cultures, and isolating different bacterial species, as well as some of the microscopy methods, some of the basics of microscopy that I discussed. But before we get into we'll start with the culturing. And before we get into actually working with the plates, I thought it'd be neat to show something about the Winogradsky columns that everyone is going to be building. So let me just change the camera here. And this is our Winogradsky column. This is what you're going to be building. I mean, get this out of here. That's for later. And you'll notice that what I've done is I've actually inserted wires into it. So there's a wire that's going down into the bottom area of the column. And that's where the copper is exposed, and the rest of it's insulated all the way up with, the insulating, the plastic. And then I have another wire that's just in the water area, so kind of the photosynthetic area. And this has been sitting in the sun in my office during the day. And you see I have a voltmeter for measuring voltage spending. So, for example, if I hold on to the electrodes of the voltmeter, you can see that the numbers fluctuate a little bit. That's just from, ordinary human voltage. We do have a little electricity in us. So it fluctuates a little bit. But either way, it stays pretty low. Maybe if I rub it a little bit, we get up to 30 or so. But what I can do is I can touch the two wires here. So if I put, actually it's more, yes, I do like that. And then if I touch the other wire, we actually get up to 200, and that's about 200 millivolts of voltage. See how much current we get. So if we look at amperage, and this is going to be in microamps. So if we do like that, no, not so much this time. We had a bit more last time. We actually had 23 microamps, but you can still see that there's a good amount of voltage that we get out of the Winogradsky column. And I asked everyone why that might be. And I think I got a fairly decent answer because we essentially have in our column, we have an area down at the bottom that is anaerobic, area up here that is aerobic. And this is where all the photosynthesis is occurring, and it's producing oxygen up here. But then as we go down, the oxygen concentration gets less. But we have higher concentrations of compounds like hydrogen sulfide, so Svalbardvätsgas, I think it's called, methane, and Fe2, so reduced forms of iron. And reduced, of course, means that there are more electrons in the shell of the atom, of the iron atoms. And that's why we see the voltage difference, is because what we have here is a rich supply of electrons down at the bottom, but we have up top a rather electro-hungry oxygen. So we have this potential that forms in the column. And this is the activity of the microbes. So we have photosynthetic organisms up here that are producing all the oxygen that are going to suck in electrons. And then all the anaerobic organisms, because they can't use oxygen, they're using other forms of different compounds and reducing them, so adding electrons to them, so that you have very electron-rich compounds down in this part of the Winogradsky column. And I mentioned that there are actually organisms that take advantage of this, where I think they find them mainly in marine sediments. You'll have cells that are growing in the anaerobic area, but they still need some place to put their electrons. They'll actually have a thread that they'll build up through the sediment that conducts electricity, that it conducts electrons. So that way they actually can do the electron accepting, because we're using oxygen as an electron acceptor, right? So what these organisms do instead is they run a thread up here, and that way they can actually have the electrons flowing through them and then going up here. So they can use that potential to generate energy when they respire. So I thought that was a rather interesting demonstration. That, you know, you can basically make a battery out of your Winogradsky columns in the end. And then what else? Yeah, what we went through after that were just some fundamentals on how we culture and isolate organisms in the lab. And I just wanted to show some of the practical aspects because, of course, we have to work aseptically, meaning that we have to work in an environment that's sterile. And to do that, I'm in the culturing lab right now, and we utilize, show this. So we utilize these guys. So this is what's called a laminar flow hood. And essentially what it is a giant filter and fan, and it's blowing air out and making basically a curtain of air that keeps anything that's flowing around out here in the lab away from your workspace where you're actually doing the culturing. So practically what we would do, you're simply just turning on a large fan, and these units unfortunately don't have it, but they often have UV lights to sterilize your work area as well. And then as you're working, you simply have this positive pressure of air coming out and keeping your environment sterile. Of course, oh yeah, I forgot to mention. So air blowing at you, you probably have to have a filter. Yes. So there's a big HEPA filter in this area as well that removes all particles. So you let it run for a while, then you have clean air blowing through with probably a couple of particles, but very few in comparison. And then of course, we're always sanitizing with either ethanol, quite often just ethanol, or bleach solutions. I forgot to mention this actually in my talk. One of the reasons we use ethanol mostly, and it's a 70% mixture, and you've probably seen that before, but one of the reasons you use sort of higher percentages of ethanol is that you really want to make sure that you have a solution that goes all the way into the tiny little microscopic cracks that are in your workspace. So you don't want a solution that might be something like water, which is, essentially kind of polar. I mean, ethanol is polar too, but to a much lower degree. So you really want to make sure that you have a solution that is getting into all the small cracks. And then that's also the thing about the UV light. UV is good, but sometimes you can still have small crevices, microscopic ones, that the UV light doesn't quite get into. So when we're working in an aseptic technique, in sterile conditions, we try to control as much of the environment as we can to avoid contamination. Yes, so I think what we did after that, well, I know what we did after that, was we went through a little bit about how do we isolate organisms? How do we get single colonies? I remembered from the lecture that that's what in the end, you're trying to seek out and get a single organism and a culture of that single organism with no contamination and no other organisms present. So one technique that's used is, it's a very old technique, but still fundamentally used, is essentially what's called dilution streaking. And dilution streaking is a way of diluting out all the bacterial cells that you might have in anything, a suspension, either of cells or maybe even an enrichment culture, and then diluting it out until you're just scraping out a single cell into a section on a plate, which hopefully it is a single cell that will then turn into a colony, and then you can actually manipulate that when you want to do other tests and confirm that it is a single organism. So I just wanted to give a demonstration about dilution streaking very fast. Let me just set up here. So let's see here. I'm gonna stop recording. Okay, I'm back, sorry. Yeah, so dilution streaking. Let me change cameras here. So we should be able to see the workbench that I'm working in. Hello. So we have our inoculation loop, of course, and this is essentially just a platinum wire that we can use, platinum, so it avoids any kind of corrosion when we're working with different medias at different pHs or other problems that you might have if you're working with a type of metal that is corrosive. People often use plastic ones that are disposable, but it's kind of nice to reduce your plastic output. And before we start, we have to sterilize, of course. And for the demonstration purposes, let's pretend that this is our culture. This is just a dye, but it'll show kind of how I'm working on the plate. So if we have our agar plate, the first thing we'd do, of course, sterilize our needle, and we get it to the point where it's glowing red. So just burning the heck out of everything. And of course, you have to let it cool so you don't cook all your microbes. Or in our case, our pretend microbes. But either way, of course, we'd be working under the fume hood. Let me turn that off. There we go. So we would then fill up our loop with the inocula. You can see it's tucked into the loop there quite nicely. And we start off by streaking it out into one section of the plate. So we're just going through and then streaking it out, covering it. And then we sterilize our needle again. or a loop rather. Waiting for it to cool. And then if you see here, this is where our start of the culture is. This is where we started the streak. We then turn it. And after it's cool, after our loop is cool, take one swipe through and then zigzag it. Put it back. And it's a bit hard to see here, unfortunately, because the dye is not so bright, but I'll draw it on there anyway. So we have a section where we've gone in this direction, and then we can turn it. And then once we know it's cool, we take another swipe through, and then we fill in the rest of the blank area on the plate. So we've diluted it at each streak. So it's more concentrated here, but then once through and then a dilution, sterilize it, once through and another dilution again. So what you wind up with is a pattern on the plate, of course, is something that starts off here, goes to there, and then goes to there. So 1, 2, and then 3 levels of dilution. And in the end, what we should have, if we look at a real culture, this is actually E. coli, Escherichia coli. It's a common gut pathogen, well, it can be a pathogen, different variants, the common gut bacterial species. And I streak these out, and I'll just put it down there. I think it's easier to see. You can see here that we have, this is the area I started in, higher concentration. But here we can have individual cultures. So these are single colonies that most likely derived from, hopefully, single cells. It could of course, be a clump or two, 3 or 4, but that you test later on as you continue to work with it. But for the most part, this is how you get a single isolate when you're working in culture techniques. So that's just the basis of general culturing. And of course, there's many other ways and other different technologies for doing even more advanced culturing, like what I went over in the morning part of the lecture. But it's kind of what I said, that you wind up having microbiological methods are really a mixture of rather old techniques and new techniques, and still nothing replaces doing these types of techniques if you really want to isolate the organism. Yes, the single cell techniques, of course, are great. And especially if you can't grow them, then you really don't have much of a choice. But you still need to be able to grow them if you want to test any hypotheses on what they are doing and how they're doing it. So, like with many things, we're in microbiology, you're using a combination of old and new techniques. So that was pretty much the basis of it. And I think we can now move to the microscope. So I'll pause here. Welcome back. So now we're in actually the microscopy laboratory. And I thought we'd go over a bit some of the basics of microscopy. So here we have kind of a classic model. This is a Zeiss, made in Germany. And I think the best way to start in talking about microscopy is essentially what we're doing is we're manipulating light and how it refracts and how it behaves and how to, and its wavelengths and what to do with wavelengths, basically, in order to see specimens that are A, too small to see with the human eye, and B, that are essentially transparent. There are quite a few organisms, they produce a color or something that makes them visible, especially when they grow in large bunches. But quite often, most organisms, they're completely transparent. you can't see them unless you have special ways of getting either staining them with a different color, or you can use light in and of itself and just manipulate it. But just to point out some of the most important parts of the microscope first, is we of course start with the light source down here. And we have an iris to control the amount of light that's being let through the aperture. And up here, this part, this is sort of the first lens system that the light passes through, and this was what is called a condenser. So it's taking the light that's coming up through here, through our iris, and then it's being focused onto the plane where our microscope slide is, where our specimen is. So all the light that's coming here is then being focused down to this one small spot. And one of the things that's important for this lens and for microscopy in general is that if we light up our specimen and we look at it under the field, we still have a field that we can look in different areas of. And we want all these areas to have the same illumination, or the same level of light that's hitting it. So what we do is we focus this in a certain way to get what is actually, it's a term called Köhler illumination. And what we're doing is creating a completely flat field of light in which we don't have any dark spots and we don't have any aberrations to make the entire field the same. So we can more easily see what's, if it's a larger structure, we can more easily see and differentiate across different parts of our structure over the whole field. So there's no differences. But also in this condenser, there are other types of apertures. So specifically defined holes that allow the light to go through. And this microscope is using what's called phase contrast to be able to see specimens that are without stain, färdiga ämnen, without stain, are completely invisible. And I think the best way to describe it, what we have here in this area, there is a ring that allows the light to pass through. So there's a big dark spot in the middle and then a ring. So it kind of looks like a solar eclipse where the moon is just a little bit smaller. And you got this ring of the sun around it. And then of course there's a larger ring around that. So it's just letting light through that one ring. And as the light's passing through that, it then goes through our specimen. And the specimen, of course, if you have this flat, if you have your microscope slide, it's such, and they're all stuck together, unfortunately. There we go. So in our microscope slide, you'll of course have cells, but then the cells, they're not all completely clumped together. There's going to be other stuff. There's going to be water there, right? Or another type of media. And as the light is passing through the media versus through the cells, it changes its phase. And the phase being, light is a wave. It's a wave and a particle. So, but it behaves like a wave. So you get light passing through, and then of course it's passing through at a certain wavelength. But then as you wind up having it go through media, maybe it's vibrating at one wavelength as it's going up through. But then as it goes through the cell, it's held back a little bit. So you get a bit of a difference in your wavelengths. And then you can actually pass it through another type of filter that puts it back just a bit more. So what you end up with is you have some wavelengths that they're vibrating together. So they kind of amplify each other, but then you get other ones that are canceling each other out. So you have interference, constructive, which is amplification, or destructive, which is canceling out. And by manipulating these differences in the wavelengths, you can then see your specimen without having to stain it. And there's a filter in each of these. And you can take one off here and show you. So if we take off the objective lens, you might be able to see as I hold it up to the camera. There we go. Yeah. You see that there's a ring in there? So that ring is filtering the light that's coming from the diaphragm here, or this annular ring as it's called, this ring that looks like an eclipse. And these match up optically. So that's how you can manipulate the light so that the light that's passing through just that ring, and then it goes through a specimen and the wavelength gets shifted a little bit, and then it passes through this dark ring in the objective and it shifts it just a bit more. And then you wind up with either an interference, which is how you can actually see the cell and different parts of the cell, or sort of an amplification. So you get this sort of halo effect. Some parts are brighter and the background is a little bit darker than normal. And then of course we have, as the light is passing up through here, we then have our, what's called our ocular eyepiece. So down here, these objectives, they can magnify objects. Let me put that down. You have this objective is listed as 16 magnification. But of course, the magnification that you see is multiplied. It's a combination of this lens and the ocular lens, what you're looking through. And this is actually 10 times magnification. So if you combine it, you have a total of 160 magnification. And then you have various different objective lenses that have different magnification strengths, 10, 20, 40, and some of them go up to 100. Those are a bit more specialized. They require oil on the slide because of the way the light is bouncing around. But I won't get into that. This is just a bit of an introduction. But what I can do is show what the difference is between these different types of phase and what the and ways of looking at invisible specimens on the microscope. So let me just pause this here. Hello again. So now we're back. And this is actually a bit more of a fancy microscope that has several cameras on it, but it's still the same basic setup. So we have our light source down here, and then we have our condenser and our different optical tools for looking at invisible specimens, invisible cells, otherwise transparent, and our objectives, and then a whole bunch of other bells and whistles. But I thought what I could do is show you a bit what the difference is. I don't know how many people, there weren't so many people that have actually seen differences in microscopy and what we can do with these different techniques for contrast. But let me switch the camera. And here we're looking at, this would be an ordinary, what we would call bright field. You can already see there's a few organisms there that do show up. They're quite large, but for the most part, it's really hard to see any detail in this picture, no matter how much I try to focus it, with the exception of those much larger organisms that are swimming around. And they're pretty big, so they're actually still doing a good job of blocking the light so we can actually see them. But the difference being, if we actually put in phase contrast, you'll see that it's much, much different. And you can see those organisms in much greater detail. And those guys swimming around are called cilia, or sorry, cilia. They're ciliates, probably a paramecium of some kind. This is the name of the group. And they're basically covered in very small hairs that help them swim through the water. And you can see that the way that they look, the way everything looks, you have a darker background, and then you get these sort of much lighter rings that are around the organisms, and then that are sort of making the outline of the organism. So this is an example of how the change in the phase of the light waves is giving our contrast. So wherever it's brighter, we're seeing a constructive interference. But wherever it's a bit darker, it's destructive interference. So the background is a bit darker. And also the cell structures, different, or if it's a protozoan, different organelles within the cell show up a little bit darker against maybe a lighter background. So it all depends on how the light is passing through our cells that we're looking at. One other form of contrast that I pointed out, and by the way, we went on a digital safari where we saw quite a few very active ciliates and paramecia. And I was hoping I could find a few other organisms as well. So we'll search a little bit here, but I'll pause the recording until I find something. But I do want to show this. We have another type of contrast called differential interference. And the difference between that and our phase contrast is simply that we're using polarized light in differential interference. And that gives us a slightly different type of contrast that actually makes the cells look 3D. And I can show that here. If we switch over. Darken it a bit. So once again, we're playing with the vibration of light. And ordinary light vibrates in all directions at once, right? So, if we're doing like this, what we really mean is as a light wave is coming towards your eye, it's vibrating in all directions. But polarized light is only in one direction. And then of course you can have, it's like sunglasses, polarized sunglasses. If you're to take two sets of polarized sunglasses, you're cutting out light that is vibrating, I think, in this direction, if I remember correctly. And if you're to take another set of sunglasses and hold them perpendicular, you cancel out the light completely. So it's another way of manipulating light so that you have a type of interference that either makes your image dark or brightens it up. And in this case, when you cross polars, you're not doing any constructive interference, but you're just allowing the light, when it's crossed, you're just blocking out the light completely because it's only allowing in one direction. But what we're doing here is we have light vibrating in one direction, but that is, as it goes through our specimen, it can shift a little bit because it's being refracted. And then you can have another crystal, another filter up here, another polarizer, and you can change the angle of that. And depending on how the light is shifted, you can get more or less light being let in, depending on how the light is being bent by the specimen itself. So once again, you're manipulating the light so that you can actually get contrast. Let's see if I can focus that a little bit here. You can go up in magnification too. So right now we're at 2200 magnification, but when we go up to 40, and then we adjust our polar a little bit back in this direction. Now we can start to see a bit more of the 3D effect as we go through. And differential interference is really good because if you remember, we're working at very small scales here.
And sometimes the cells you're looking at they can be quite thick. And with phase contrast, oh, there's something, a diatom. See that little oval speck in there? That's just about in the middle of the screen now, underneath that clump. That looks like a diatom. So those are interesting organisms. They're protozoans that produce these silica shells around them. And you can have very large deposits of these. Once they die, they leave their shell behind. And they're actually quite useful in industry. I mentioned this to the class, but today I had a fun experience with that because I went to the dentist, and as they were polishing my teeth, they were using diatoms, basically. It's a combination of diatomaceous earth and a few other things that they add, but it's a way of polishing. It's an abrasive because it's silicone, really, really fine sand. But getting back to what I was saying about phase contrast and the differences between that and differential is that you can get you can see structure in thicker specimens with differential interference contrast. This type of contrast, phase contrast, maybe you can switch over that, you might get a better idea. It can be a bit, not a great example, but it can be a bit more difficult to see structure in all different planes of light. Because remember, if it's a thick specimen, you're only looking at one plane at a time, but it's still thick. So maybe there's something interesting up here, but maybe there's something interesting just below it. But it's blurry and you can't really see it. You have to go back and forth. You can get a slightly thicker one, slightly more detailed view of the structure when you use different types of contrast. I might also add there's one other type of microscopy we can use if we really want to see organisms that are otherwise largely transparent. So if I go back here, now let's look at our phase contrast just to get an idea. And you might be able to see a few things swimming around in there, meaning very small ones. And the smaller ones are, of course, going to be bacteria at this scale. But what we can do is we can actually do something called bright field. And if we focus it a bit better, it actually becomes much easier to see organisms that are swimming, that are quite small. So what we're doing now is taking the light and basically angling it in a way that the only light you see through the lens is that which is bounced off of the organisms and comes through into your line of sight. So the background is dark, but then everything else that's moving or that's reflecting and bending light lights up. So this is called dark field microscopy. And it could be another way of contrast, another form of contrast in seeing organisms that are otherwise, if we go back to the bright field, completely transparent, or at least extremely difficult to see. I'm going to pause here because I have one more example to show. So this is what I wanted to show. So here we have a culture of cyanobacterium. And cyanobacterium, as you might have remembered from the lectures, it's quite special. It is photosynthetic, but one of the other things that it does quite well is it fixes nitrogen. So it converts nitrogen gas that is in the atmosphere and that's unusable to organisms into a form, ammonium, that it can use, organisms can use. And to do this, it has enzymes, an enzyme called nitrogenase. But the thing is, the nitrogenase does not work when oxygen is present. Oxygen, in fact, deactivates the enzyme, it kills it off. And that can be problematic. If you're a photosynthetic organism that produces oxygen, but you want to also be able to utilize the nitrogen in the atmosphere to build up your biomass, your proteins, and all the other macromolecules that a cell needs. So what it does, the cyanobacteria, it produces these structures in which nitrogen fixation takes place. You might be able to see it. It's now at the very center of the screen of these round structures, these cysts. And here is where the nitrogen is fixed. And these structures keep the oxygen out. So that way the enzymes are not killed off. So that way you have cyanobacterium that are capable of using carbon dioxide gas from the atmosphere, as well as nitrogen gas from the atmosphere. And that's why quite often these are the first organisms that you'll see colonizing. Let's say if you have a volcanic eruption or something and it completely kills everything off and there's nothing but bare rock. These organisms are quite good at settling in places and colonizing places and thriving there. One other thing I wanted to show you as well with the cyanobacterium is, of course, they're photosynthetic, therefore they have chlorophyll. And in microscopy, we can actually use fluorescent light to also visualize molecules that fluoresce. And just a quick explanation of fluorescence, when you have an atom and then you strike it with a particular wavelength of light, there are some molecules where the electrons in the atom, they jump up to a different level in their energy, and then they want to go back to where they came from. But in order to do that, they have to emit light to kind of release a little bit of that energy. So essentially you have one wavelength of light coming in, one color. Let's say, well, usually it's going to be the rather short wavelengths, but you have one wavelength coming in, and then you have another wavelength coming out. And this is how molecules fluoresce. So chloroform does that. And when we look at it under fluorescence, let's see if we can get it to work. I'm going to pause this until we get it working. And there we have it. So now we see the chlorophyll in the cells lighting up when it's being hit with a fluorescent light. Let's see if we can play around with that a little bit. No, that's the best one. And it fluoresces red. So it's another way of visualizing cells. Here, these organisms sort of provide their own fluorescent stain, but we have many different compounds that we can add in, we can stain cells with, that also give off different colors when you hit it with different wavelengths. So there's a variety of different tools that we can use to visualize cells in this way as well. Yeah, okay. That was the basics of what we covered. And there was the additional field excursion video that we also went through a little bit. So you can take a look at that, as well as some of the other videos that I put up from my colleagues that are working more with fungi and fungal species. They can explain it far better than I can. I mainly work with prokaryotes. So hopefully this covered everything. And if anyone has any questions, please email me. Okay, thank you.